Hi everyone, Miss Portelli here. This set of notes is our last one on dihybrid inheritance. Specifically today we're going to be looking at something known as a dihybrid test cross and how we can use a dihybrid test cross to determine whether two genes are linked or independent of each other. So let's take a closer look at that now. In our previous videos we spoke about the difference between independent and linked genes, independent being two different genes that code for different traits on different chromosomes, and linked being two genes for different traits that are on the same chromosome. In our previous video we also looked at examples of independent dihybrid crosses where we had two heterozygous individuals for both traits being crossed together, and we saw that we usually produce a 9:3 to 3:1 ratio when we combine two heterozygous individuals for independent or unlinked genes. If you need a refresher on that, then I'd suggest you pause this video now and go back to rewatch our previous video on that. So what we're going to look at from this point forward is looking at using a dihybrid test cross, so a specific type of Punnett square or a specific type of breeding between two individuals for the dihybrid traits that we're looking at. And depending on the ratio that we will see in the offspring, that should hint to us whether we're looking at two independent genes, like the example on the left of the screen here, or whether we're looking at two linked genes like we're seeing on the right of the screen here. So I'll talk to you about those ratios and what this dihybrid test cross looks like, and then hopefully you'll be able to apply that to some new examples yourself. So when we're performing our dihybrid test cross, unlike our monohybrid test cross we've used previously, in our dihybrid test cross we know with 100% certainty the genotypes of the two parents that will be involved in this dihybrid test cross. We know that we will be using a heterozygous individual for both traits, and we will be crossing that heterozygous individual for both traits with a homozygous recessive individual for both traits. So if we keep referring to our P example that we used in our previous video, we would be crossing a heterozygous individual for yellow and smooth Ps. Okay, our yellow being the capital Y lowercase y, heterozygous for that, and then the smooth P is the capital R lowercase r, heterozygous for that trait. We would be crossing that with our homozygous recessive, our green wrinkled P, green phenotype coming from the lowercase y, and the wrinkled phenotype coming from the lowercase r. So if we think about these two parents that we would be using in our dihybrid test cross, we would need to think about what gametes they would produce before we could potentially fill out a Punnett square and predict our phenotypic and genotypic ratios. So let's have a look at that now. So if we look at the possible gamete combinations for our heterozygous individual, that's the yellow smooth P, these are the combinations that we can potentially get. So you can see, let me just change my color of pen, that we can get a combination with the capital Y, capital R here. We can get a combination with the lowercase y, capital R here, the capital Y, lowercase r here, and then both lowercase letters are our fourth gamete that this P can potentially produce. You can probably see where this is going next. If we were to look at our green pea plant, our green wrinkled pea, what would the potential gamete combinations be for this green wrinkled pea plant, our homozygous recessive? Well, this pea can only really pass on one potential combination, and that combination is lowercase y, lowercase r. So it would essentially, instead of having four different combinations, those four combinations that are produced would be identical, and it can only pass on a lowercase y and a lowercase r. So with that information, we can now set up a Punnett square and predict our phenotypic and genotypic ratios using that Punnett square. So let's have a look at that now. So we set up our Punnett squares as usual, where we have one parent's gametes across the top here, where we've got our heterozygous for both traits, and then we have our second parent with the gametes down the side here, and that would be our homozygous recessive. And you can see that our homozygous recessive in each of those 4 boxes, we've got the same gamete combination that is being passed on to the offspring. So when we start to fill out our Punnett square, so for example, if we fill out that first box in the top left, we're going to get capital Y, lowercase y, capital R, lowercase r, our combination from both parents. And if I continue to fill down in that Punnett square, you'll notice that all 3 of those boxes underneath are going to give me the exact same combination, because parent 2, our homozygous recessive, can only pass on 1 combination. So to save ourselves a little bit of time, normally when we perform a dihybrid test cross, because we know we're crossing a homozygous recessive with a heterozygous individual for both traits, we tend to ignore that bottom 3/4 of our Punnett square to save ourselves a bit of time in filling out all 12 of those squares, and we're just going to focus on this top row here. So for the next couple of slides, you'll see that I've blanked out those bottom 3 rows because they're just a repeat of the top row. So please don't freak out if you see my Punnett square a little bit smaller than usual. So this is what the Punnett square would look like if we had filled it out. Remember, I've blanked out those bottom 3 rows because it's a repeat of the top row. So just focusing on the top row, we can see our gamete combinations that would be passed on to the children, and we can see that we produce 4 distinct combinations. So let's have a look at those combinations in a little bit more detail and what ratio that would produce for us. So just like we did in our previous video when we're looking at dihybrid crosses, we need to write down all possible phenotype combinations that will be produced from this particular cross, and then we need to count how many offspring would be produced showing that phenotypic characteristic so that we can have our ratio that we're looking at. So the 4 different phenotypes that we can see in our cross here are a yellow smooth pea, a yellow wrinkled pea, a green smooth pea, and a green wrinkled pea. So you can see those images I've just popped on the Punnett square there to help you identify which genotype is going to give you which phenotype. And you can see that we've got 1 of each. So when we fill out our numbers or our ratios for our phenotype, it's going to be a 1 to 1 to 1 to 1 ratio. Okay, so 1 yellow smooth pea, the first box here, 1 yellow wrinkled, oh, not the second box, sorry, let me rub that out. That should have been the third box. The green smooth pea is the second box, and then the green wrinkled pea is our 4th box there. So we've got a 1 to 1 to 1 ratio. Previously, when we were looking at examples with dihybrid inheritance before we did test crosses, when we were looking at 2 heterozygous individuals, remember we looked at a ratio of 9 to 3 to 3 to 1. So that is a different type of cross. It is not a dihybrid test cross. The one we're looking at on our screen today is a dihybrid test cross. And if a dihybrid test cross is performed and you generate a ratio of 1 to 1 to 1 to 1, which we have on our screen here, that indicates to us that these 2 genes are unlinked or they are independent of each other. So the gene that produces the colour in peas is on a different chromosome that the gene that is responsible for the smooth or wrinkled texture of the pea is located on. So the gene for colour might be on chromosome 3, and the gene for being smooth or wrinkled might be on chromosome like 17 or whatever chromosome it is in peas. Okay, let's step through that and add on a little bit more now. Okay, so we've just looked at the example of the peas, looking at the gene for the colour of the pea and the gene for the texture of the pea being smooth or wrinkled. And we showed using our dihybrid test cross where we crossed a homozygous recessive individual for both traits with a heterozygous individual for both traits, and we found that the offspring phenotypic ratio was a ratio of 1 to 1 to 1 to 1. So the picture that you're looking at on the screen right now is an example of another scenario where we're looking at a heterozygous individual here being crossed with a homozygous recessive individual for the two traits here. Doesn't really matter what the A's and B's represent, more so that they're representing two different genes. So gene A and gene B, they have performed a dihybrid test cross, and we know that it's a dihybrid test cross because it's a homozygous recessive individual, so the one on the right here being crossed with the heterozygous for both traits, the one on the left here. We can then see the phenotypic ratio in the table here, which is showing 25%, 25%, 25%, 25%, or another way of saying that would be 1 to 1 to 1, whoops, to 1 to 1. Okay, so the 4 ones in the ratio. And that information, reading that from a dihybrid test cross, tells us that these genes are unlinked. So they do not appear on the same chromosome. An image of what they might look like in parent 1 is shown down here, our heterozygous individual, and an image of what they might look like in parent 2 is shown on the right here for our homozygous recessive individual. So the takeaway message for this side of the screen is that when we perform a dihybrid test cross, if two genes are independent of each other, we are expecting a phenotypic ratio of 1 to 1 to 1 to 1, or you might say it as 25%, 25%, 25%, and 25% for each of the 4 phenotypes that we would see in the offspring. But the point is that they are all relatively equal in frequency. Now the next example that I'm going to show you is the one for if a dihybrid test cross is performed and two genes are in fact linked, so they do appear on the same chromosome. So let's just pop that on the screen. So if our phenotypic ratio for a test cross varies from the 1 to 1 to 1 to 1 ratio, we're going to assume that two genes are linked. So if we look at the example that's on the screen, on the right hand side of the screen now, and I might just change my pen color so that you know I'm talking about a different one, so I'm going to be drawing in purple now. We're still crossing a heterozygous individual with a homozygous recessive individual for two traits. So we can see that the heterozygous individual, circled in the purple on the left, capital E lowercase e for one trait, capital F lowercase f for the other trait, so that making them heterozygous. And then we've got our homozygous recessive with 2 lowercase e's for the E gene and 2 lowercase f's for the F gene. When we draw up our Punnett square, remember we only have to do one line of the Punnett square because our homozygous recessive individual, our parent 2, is going to have the same the whole way down if we were to fill out all 4 rows of our Punnett square. But the important information on the screen that we're looking at is our phenotypic ratio that would be generated at the end of this cross. So if this cross had been performed in real life and those offspring had actually been counted up, we would have found that 44% of the offspring would show this particular genotype here, so they would be heterozygous for both traits. 44% of the offspring would show this genotype here, so homozygous recessive for both traits, and then 6% each for the other 2 options, where they're homozygous recessive for one trait and heterozygous for the other trait. So it's really clear to us looking at those numbers on this particular Punnett square that we don't have 25%, 25%, 25%, 25%, or that 1 to 1 to 1 to 1 to 1 ratio. Okay, but what we need to understand is why is this happening? So we're going to have a quick chat about what this means for linked genes on the next couple of slides, and then we'll have a look at an example of this put into action.